Okay, so today we're going to talk about chapter 3 of Abbas, which is going to talk to us about leukocyte circulation and migration into the tissues. Okay, so leukocyte homing and recruitment can be broken down to a few different primary roles that we're going to try and understand. One is how neutrophils and monocytes actually get to the site of infection. But then two, how the lymphocytes first go to those secondary lymphoid organs. Remember, they kind of have to be trained. They're naive until they actually see the infection. They're the adaptive ones. And then they actually have to leave those organs and get to the site of infections. The neutrophils, the monocytes, that innate part of the immune system doesn't need that. They can go straight to the site of infection. So here's a broad scheme of exactly that. So here we talked about earlier in the Sympyric text that as the neutrophils and the monocytes are rolling along in our blood vessel, what they have to do is stop at the appropriate place and then squeeze through the blood vessels to get to the site of infection. Right? It's more complicated for TMBs because they start out as naive until they actually see a specific infection. And they do that in the lymph nodes. Remember, these lymph nodes are kind of like dating bars. So they need to get here and spend a lot of time in here to try and see a antigen presenting cell presenting an infection and an antigen, and then actually activating that cell. And only then does it leave and then go through the same thing that the monocytes and neutrophils do, meaning it has to get, or some of them at least have to get to the site of infection, stop at the appropriate place, and then squeeze through the vessel walls. Okay, so a lot of this is done through a series of adhesion molecules, selectins and integrins, which bind to selectin ligands and integrin ligands. These are outlined on table 3.1, which I think is an excellent table to know. The adhesion molecules, such as the selectins, they initially have they're the initial low affinity adhesion. So we think of selectins as slowing down the cells. There are P-selectins and E-selectins, and these are found on the endothelium. Therefore, the selectin ligand that they bind to, which in this case is Lewis X, is actually found on the cell that needs to slow down. So the selectins will slow, the integrins will stop. The selectins can either be on the leukocyte themselves or the endothelium. It's P and E selectins on the endothelium. E selectin for endothelium. The L selectin is on leukocytes. L for leukocytes. They bind the sialomucin on the endothelial cells by the peripheral node addressin. Both of these get increased in inflammation, and that's what you want. So if you have local inflammation, that's where you want these cells to stop, slow down, and go. If you have an infection on your toe, that's where you want everything to move, but you don't want the cells to necessarily be slipping out in your hand. So it's these local areas of inflammation will increase the expression of these things, allow for that interaction and things to slow down. And then once it's slowed down enough, then you get integrins, which actually mediate the adhesion. The selectins are that low affinity slowing, integrins mediate adhesion. So there is LFA1, which binds to ICAM1 on the endothelium. There's VLA4, which binds to VCAM on the endothelium. There's MAC1, which binds to ICAM on the endothelium. And then there is one called CD11C, CD18, which is also called CR4. So to me, the most confusing part of this, again, is not conceptually, right? We can get that selectins slow down, integrins stop, and then that allows the cell to slip through. Problem in my mind is that there's so many different names for all these different integrins and selectins. That makes it very difficult. Things that I would point out, though, are like LFA1, because if someone has an immune deficiency, leukocyte adhesion defect. They're missing CD18 adhesion molecule, so you would not have LFA1 and MAC1. That's the type of context I'd put it in as you're trying to decide what to remember. The other thing I'd point out, LFA1, MAC1, and then also this CR4. So these two integrins also happen to be complement receptors. I would try and keep that as well. There's one specific one, alpha-4 beta-7, which binds to MAdCAM1. It's found on the gut endothelium, and I call that one out specifically because there's a biologic that uses this as a target. So whenever you see something, again, that has this high clinical relevance, I would pull it out as one to know. So that's why I pulled out alpha-4 beta-7. Okay, along with the selectins and integrins, which slow and stop cause that adhesion, there's something called chemokines, and chemokines are cytokines that cause leukocytes to move. There are several different kinds, including CC chemokines, which support monocytes, lymphocytes, and other leukocyte migration. And there's CXC, which support neutrophil migration. And then there are C and CX3C. This just shows the pattern of the molecule, kind of that structure. I think overall less important. But what you're going to see are things like CCR7 and CXCR3. So this is just kind of a setup to that. I don't think you need to remember too much more. They bind to a 7-transmembrane G protein coupled receptor. So this is just a receptor that can be identified because it goes through the membrane 7 times, the cell membrane 7 times. So it kind of goes up and down and up and down and up. And then, of course, has a part on the outside to bind to, and then something on the inside that allows it to signal. So don't let this scare you. It's long, but it's just a certain type of receptor. A lot of this is on table 3.2, which has some relevance. There's a lot on this table, but I've tried to pick out some as the text that are highlighted in the text that are really important to our understanding of what's happening. Okay, so chemokines cause the recruitment of leukocytes from the blood. They stimulate the movement towards a chemical gradient, which is a phenomenon called chemokinesis. They allow the development of the lymphoid organs, and they regulate traffic of lymphocytes through the peripheral lymphoid tissue. They also cause the migration of dendritic cells to those lymph nodes using this CCR7. So you'll get this all in 1 second. So the selectin-mediated rolling is upregulated at the site of infection, which we discussed. The leukocytes move close to the walls due to vasodilation and slowing of the blood flow. So you get vasodilation, you get slowing the blood flow, you get that low affinity connection, and you get this repetitive binding and detach, bind. So it actually starts rolling around on the outside of the endothelium. And then the integrin, so chemokines from the endothelium bind to the chemokine receptors on the leukocytes. It causes enhanced affinity for the integrins, and then you get this stable adhesion. This is not just one point of contact for a cell and endothelium, it's many. So it will cause the cell to actually kind of flatten and spread out along the endothelial surface, and then it allows it to transmigrate between 2 endothelial cells. So the selectins, you get this slowing. Because of the inflammation, you get that reduced blood flow. It goes to the outside of this the vessel. You get that interaction because you've had upregulation of the whole system due to the inflammation. On-off for selectins, it starts rolling, and the integrins actually bind it, flattens it, and then it goes right through the endothelial cells, and that's how it gets to where it's going. So here is a good example of exactly what was just discussed. So you have these low affinity integrins like LFA1, and here are the integrin ligands. And In pink, in this case, it's ICAM1, which is the ligand for LFA1. So you start to see this adhesion, but you also get the chemokine and chemokine receptors that is going to allow this to kind of have that secondary signal that this is where it should be slowing down. There's many, so this is eventually going to start to flatten where it can then sneak out through the vessel right here. Here is a bigger picture of everything that we just discussed. So here you have blood flow through the endothelium, but you have the site of inflammation. So you're going to get an increase in selectin and integrin ligands across the endothelium here. So you have your leukocyte that wants to come. It's going to start binding with these selectins and low affinity, and it's going to bounce, bounce, bounce. But it's eventually going to have the integrins bind with the integrin ligands where it's going to stop and it's going to spread out. And then it's eventually going to be able to just slip through, as we can see over here, between two cell walls to the site of infection. I'm going to stop there for a second. Is there any questions about all that?
Nope.
There are two primary immunodeficiencies that are highly relevant to this. The one that is more common is leukocyte adhesion defect 1, LAD1, and this is where you have a deficiency of CD18, which affects several, I think it was 3 of the integrins. So you don't have that adhesion molecule, so you don't have that leukocytes. You lose that leukocyte's ability to adhere. So clinically, what do you see? You see very high numbers of white blood cells in kids' blood all the time because the body knows that it needs to get white blood cells to infection, so it's making more white blood cells, but they just keep going by the site of infection. They cannot adhere, stop, and actually get to the site of infection, which is how it, one of the ways that we are supposed to recognize it are the sterile infections, right? And so you have these soft tissue infections and you biopsy them. You actually don't see those inflammatory, you don't see the leukocytes having the ability to get there. When I've seen it, we've diagnosed it in kids often because they have gotten a CBC with a very high white count and it's ignored once. We all kind of chalk it up all the time in kids, but then maybe they got a second because they keep getting sick and suddenly we say, oh, well, they're white. We've never seen a white count low on this kid. Maybe we should send flow for LAD. There's also a less common type of leukocyte adhesion defect, LAD2, and this is where they cannot express that lectin ligand or sialyl-Lewis X. So it gives a similar phenomena. It's just the molecule that is missing is different. In this case, it's the selectin. LAD1 is the integrin. Okay, so that simplified version is highly relevant for neutrophils and monocytes. They're innate and they just need to go to the site of infection, and that is it. Adaptive immune system, like T cells and B cells, is a little bit different. The first thing they have to do is get from where they're created, like the thymus or the bone marrow, and they need to get to the lymph node where they can actually then have an encounter with an antigen presenting cell like a dendritic cell. So in order for that to happen, all the lymphocytes, like naive T cells, need to get to the lymph node, but so do the dendritic cells or the antigen presenting cells that are holding the antigen. And within that node, they have to be directed to the same spots. So remember, there's T cell zones and B cell zones of that lymph node, and at the right time, it has to go to the right place. So this happens by an upregulation and downregulation of different chemokine signals and selectins to try and direct it to the right place. Okay, so in this case, when you have inflammation, you get increased blood flow to local lymph nodes. And we see that, right, because we start to see lymph nodes that are starting to get swollen, trying to direct everything in that local area to create that match between the T cell and the antigen. And then the T cell exit from the node is delayed. So always when you're healthy, the cells are going from lymph node, they stay there and they go back in, but you're not really, your body is not trying to promote that interaction between the antigen presenting cell, and the type T cell. Once there's inflammation, that changes. It's really motivated to keep the T cell in the node. So it increases blood flow to the nodes, but then the T cell exit is delayed. It gives it more opportunity for the right T cell, that one specific T cell that would recognize the antigen, to actually get activated by the antigen presenting cell holding antigen on MHC. So it's delivered to the nodes from blood via these high endothelial venules, or HEVs. So in the high endothelial venules, you get selectin-induced rolling, and then you get a chemokine-induced integrin activation. This is done by CCR7. And then you get an integrin-mediated adhesion through LFA1, and then you get transmigration out of the blood vessel and actually into the lymph node. So the address is CCR7 to get into that T cell zone of the lymph node, and it's supported by selectin, specifically L-selectin and integrin LFA1. Okay, when T cells want to leave the lymph node, they exit following something called a CYP gradient. CYP stands for sphingosine-1 phosphate. So it binds to S1PR1, which is a G protein coupled receptor. Again, don't let the lingo scare you. It's just a type of receptor. So CYP is in a high concentration in the blood. And in the node, the CYP1 receptors can be expressed on the cell surface. So then it can actually follow that gradient back into the blood. So if you want the T cell to stay in the node, what would you do? Well, you would downregulate those CYP receptors so it's not actually chasing after that CYP. Okay, so here is the system. So you have blood vessels, you have your, in this case, a naive T cell. It's going to go through using CCR7, selectins and integrins. It's going to be able to squeeze out and get into the lymph node. At the same time, through the vessels, which we learned about, through the lymph vessels, which we learned about before, you're going to have these antigen-presenting cells, like dendritic cells, that have the microbe or antigen on MHC. It is also going to be going to the same address, that T cell zone through CCR7. And that's where you're hoping to promote this interaction. When that happens, then it's no longer a naive T cell, it's an activated T cell. That's where you want it to leave. So that's where it's going to follow that CYP gradient, and it's going to go exit through the lymph vessels. It does not exit through blood vessels. It exits through the lymph vessels, but the lymph vessels, of course, eventually go back to the vena cava via the thoracic duct. So we'll eventually all get back into circulation. And then hopefully at this time, inflammation will get it, and the address will get it straight back to infection, where it can actually, using lectins and integrins, slide out of the vessel at the right place at the site of infection. Okay, so here is kind of the exit strategy. So once you have a naive T cell coming in, remember this is addressed to CCR7. It's going to the T cell zone by CCR7. So it does not have much of the CYP receptor. So it's not going to chase the CYP gradient. Once it gets activated, that's going to change. The CYP receptor is going to go up, and it is going to start chasing that CYP gradient. And those CCR7 is going to decrease. It's not going to want to stay in the T cell zone. So it's this dance of upregulation and downregulation, depending on where it needs to move, and then it leaves. Of course, to some extent, naive T cells will leave too. They're not all going to get activated in here. If it's been in here long enough, it will eventually just start increasing its expression of CYPR1 to leave and go to a different lymph node, because maybe this particular naive T cell is not needed in the toe, it's needed in the hand, so it needs to keep circulating and going from lymph node to lymph node. Okay, so here, this is actually a great table. Maybe I'm not going to go over into too much detail, but this gives the specifics of all these different interactions, specifically the ones we've talked about already. is the L-selectin for the naive T cells binding to L-selectin ligand, CCR7, I would know cold, binding to its ligand and LFA1 to ICAM1. This is going to get that T cell in activated or memory T cell, so it's going to have that CYP gradient that it follows, but it's also going to have to get to the site of infection. And when that's the case, it's going to upregulate these EMP selectin ligands, which will then bind to EMP selectin. It will upregulate CXCR3 and CCR5, as well as integrins, LFA1 and ICAM, or VLA4 and VCAM. These are two different types of integrins. Okay. B cells are very similar, but B cells don't want to go to the T cell zone, so they're not going to follow CCR7. B cells want to go to the B cell zone, so they're going to follow something called CXCR5. And then they'll leave the same way. They'll leave via that CYP gradient. Okay. So here is the migration cycle, just like we looked at before. So you have your B cells, which of course are circulating through our blood. We know it's circulating through our blood because if we get a CBC with this, we can always look at our B cells. We know they are traveling, but they need to make stops at the lymph node, just like the T cells do. So they're going to leave via the high endothelial venules from the blood vessels into the lymph node, but this time they're actually going to follow CXCR5 to get to the B cell zone of the lymph node. Once it is activated, if it's activated by a T cell that recognizes it, then it's going to upregulate that CYP gradient, and it's going to leave, not by blood vessels, but by lymph vessels, but then the lymph will eventually dump back into the superior vena cava by the thoracic duct. And then once you have your activated B cell, of course, it's going to turn into a plasmablast, and then it will largely go back to the bone marrow, chasing a whole different set of markers like CXCR4, getting it to the bone marrow. There are some, of course, that will go to, like the mucosa, in the case of IgA secreting plasma cells, it will chase something different like CCR9. Oop. That's actually it for this chapter. Let me stop for a second.